Hello, my name is Theodore Bullis, and welcome to OhmyoBio video number 1, name not final. So today we'll be talking about viruses. Viruses are really interesting things in molecular biology for a lot of reasons. And one of the most interesting things is that even though they work very closely with cells, or against cells, they're not actually alive like a cell. They're not cells. And there's a lot of reasons for this. One of them is they don't share the characteristics of cells. They don't have a cytoplasm, a cell wall, or ribosomes, although they do have a form of nucleic acid, be that RNA or DNA. And also they need a host to survive, so they need to be inside another cell to be considered a cell. But these viruses, even if they're not alive, are really amazing in a lot of ways. And the first one is that they just look cool. I mean, look at that thing. That is a bacteriophage. And a bacteriophage injects its DNA with this long needle and heel into its cell. And this is, you have to think, only a few nanometers big, or even smaller, and yet it looks like a killer death robot. And whenever I hear the word microscopic killer death robot, I know something cool's happening. I mean, it's not actually a robot, although you can kind of think of it like that. Because it mocks the characteristics of life, like an android would, while not actually being alive. And some of the ways it mocks the characteristics of life is it loves to reproduce, like every living thing. It tries to reproduce and create as many of its self as possible. And also, it evolves. And when I say evolve, I don't mean like the 50 million year natural selection evolve. I mean the 2-week radical alteration immune to vaccine types of evolve. The type of evolution that makes viruses so difficult to fight, because when you come up with a cure for them, they just fix themselves so they can't be cured anymore. So we're going to be talking about a lot of different things about viruses. We'll start with the structure. And the structure of viruses, although they're completely different, they're all so different, there are two things that every virus has. One, they all have some sort of nucleic acid, be that DNA or RNA. In the bacteriophage, there's DNA stored up here in this tetrahedron up here, and in something like the influenza virus, we've got RNA inside, and they're both protected by this protein coat. A lot of this virus here is made of protein, especially this area up here, and if you see here, that's all a protein coat. There's also, in some, including influenza viruses, a lipid bilayer. A lipid bilayer is just two layers of lipids organized to protect the RNA and DNA even more. So viruses, their main goal is to inject their information, the genetic information, into the nucleus or just the DNA or RNA of another cell so that that cell can build more of them and they can reproduce. So there's a lot of different types of viruses. The two main types of bacteriophages and those that invade animal cells. But as you can see, even among things that involve humans, like influenza and HIV, they look somewhat similar, and rabies. They look a bit similar, but they're not actually the same. They just have a kind of similar, like the spikes on the outside of a ball. But as you see, rabies is more elongated. Adenovirus is more of like a couple of pyramids stacked on top of each other. And bacteriophage looks like, like a droid spider. So, what we're talking about first about influenza virus. Remember how I was saying that viruses evolve? Well, that's really important for understanding why viruses exist, how they do. Because a virus, influenza has to get into the human cell. And as you can see, a cell for human has has its DNA stored inside the nucleus, inside of a cell wall, inside the sea of cytoplasm. And that can be difficult to infiltrate. So the influenza virus has a really interesting technique when it uses something called hemagglutinin, or hemagglutinin, in which those are these spiky kind of things, the red and blue stuff on the outside, that act as keys to the lock of the semipermeable cell membrane. See, cell membrane doesn't want to let viruses in, but when these things get to the surface, they kind of trick the cell membrane into thinking, oh, this is something safe I want to let in. This is a protein or an enzyme that I really need to survive. And so it actually lets the virus into the cytoplasm and even protects it and lets it travel straight to the nucleus, well, it injects its RNA to the nucleus and takes control. Bacteriophages are really interesting also. As I mentioned earlier, they have a needle in here. A literal needle that they use to inject the bacteria. As you can see, bacteria cells, they have cytoplasm, and then that's the only thing protecting the nucleic information. So a needle can easily pierce through that cell membrane or the cell wall and get to the nucleic information really easily and just inject their own. And once they inject it, there's a couple different things that a virus can do. The first one is called the lytic cycle of a phage. The lytic cycle, you've got, it goes onto the cell, and with the bacteria cell, and injects its DNA or RNA, and it basically breaks up the old DNA and tells the cell, okay, your new job is not to make energy, it's not to, only in the way it can help me, it's not to do anything for your own survival, it's to start cranking out parts of my virus. And so, they start making the head, tail, and tail fibers, as you can see here, and they slowly build inside the cell and then just burst out. And this seems really effective, because if you think about it, let's say only 2 are created every time you come in, that's still doubling your numbers, and then those 2 double, and then double and double. It's exponential. But there's an actually more effective way, and this is why viruses are so scary and so efficient, which is called the lysogenic cycle, in which, after it injects its DNA, the DNA cooperates with the cell to keep it alive long enough to reproduce itself and split into 2 cells. By integrating its DNA, and then when the cell reproduces and creates a copy of the old DNA, then boom, we've got 2 cells with a viral code ready to create more viruses. And it will keep doing that until it has enough and inducts the lytic cycle one more time, and we go to what we know from back here, in which it builds up the phages and the phages break free. This is why some diseases like HIV can go undetected for a long time, because those diseases act in the lysogenic cycle, where they first build up and have this latent phase in which you can seem perfectly fine and you're healthy until the viruses break out. And this is what makes viruses so just terrifying, because they can go undetected and just completely control your cells. Think about a sophisticated enough virus could actually control your brain, and that's what rabies does to some mammals. And there's a lot of things that a virus can do because it literally controls your cells and bends them to its will. And even though they're not alive, they're even more. They can be in some ways more sophisticated than living things in the way they can evolve. And so, viruses, because they can evolve so quickly, they can evolve to adapt to vaccines and cures, and they're a very, very difficult thing to fight. Thank you, and I'll see you later.